All right, so now the second student is coming, and there are some of the questions that Scott asked might be answered. Unexpected findings.
Okay.
What was it?
Use the camera. Your button.
Oh, how annoying. So here, Rianna Soltis, she worked with us for the summer. She's going to talk about the effect of storms on ecosystem processes and the storm experiment and some of the findings.
Thank you for the introduction, Elka. As she said, my name is Rianna Soltis. It's really nice to see you, have you all here today. I'm going to be talking about the ecosystem processes in specific to the storm days that we were doing, but I'll also go over everything that we did every day. So two main things that I'm going to be talking about are storms and oyster biodeposits. So a storm is a natural stressor to an environment, and we found it was very important after all these years of Elka running these STURM experiments and her mesocosms to go over how a storm would affect an environment. So that is why we focus on that. And then biodeposits are actually oyster feces and pseudofeces. The difference between that is feces go through the digestive system of the oysters, but the pseudofeces actually they bypass that, but there still are excrements from the oyster's body. So what we want to focus on is the resuspension of these biodeposits. So as you can see in this picture, in this video, there's actually what the storm looked like is when we spun the paddle much faster than normal. It was always running. But as you can see, there was like resuspension going on. The water was pretty murky. So what that is basically any of the biodeposits, anything on the bottom, they lifted from the bottom, and that affected the water quality, as you can see clearly here. Something else I want to point out is that the oysters were not actually kept in the tanks, even though it seems like that would make sense, but we actually took the oysters biodeposits from these tanks that were on the dock. So they're separate entities from the experiment, but still very important. And then as Sean talked about shear stress, that's just when the resuspension occurs and why it's occurring, which is just an important keyword to know. So every single day we took a plethora of data. So one of the things we used a YSI instrument for dissolved oxygen, which is what Sean's using here. And then we would use a fluorometer and collect water with a cuvette, which is this tiny little thing I'm holding in this picture. So what you would do with that is we would collect water and then we would run it through the fluorometer and test if there was any algae present or any fluorescence in the water. And then another thing we did was Secchi, which I will be showing corresponding data with. But Secchi is basically how far the light penetrates into the water and if it reaches the soil, which is the sediment, which is important to know for future nutrient reasons. So for daily, and then we had daily and I would call automatic measurements. And I call it that because there was two probes in the tanks at all times. There was temperature and turbidity. So turbidity was measured by OBS, which Sean explained more in his experiment, but they were both in there and all that data was kept on this little computer right there that never moved, closed, everything. It just stayed there. It was not touched. And so we used that every day, all day. And the mixing system, which was right here, was going on for 4 hours and then 2 hours off, 24 hours a day for 4 weeks. So that was very important to watch, and the computer helped make sure that was still going. Plus we used the stepwise erosion during the two storms, which is what Sean was talking about, and how there's different levels of the shear stress going on. Another thing we did two other things every day that were very important. One other thing was we did a water exchange with filtered water. So 10% of the water of each tank was taken out. So we would put filtered seawater in there to show that in natural occurrences, water is replaced all day in like the river because it's moving water. And then we would add the 2,000 milliliters or 2 liters of biodeposits. As you can see, that's what we're doing here actually in our really gross wet lab. And so we'd add those every single day from the oysters, and we would go every couple of days to get biodeposits from the dock. So sampling days, these were big days. It wasn't every day, but we always did it on storm days, and we did it about every 3 days or so during the experiment. So we would use a sampling stick, which is actually homemade with PVC and tubes tied to it with zip ties. And we would put that in the middle of the tank, well, Sean specifically, would put that in the middle of the tank and collect like the best water that would be overall encompassing of the tank. And then Elka would pump to get all the water to come through, and we'd get it in a jug and we'd take it back to the lab, which we all did different filtrations, but the important thing that I did was filtered total suspended solids, which are TSS, or it's also called Cest, which I will be referring to a lot throughout the next couple of slides. And we did this with prepared filters, which looked like, which is what Sean's doing over there. He's preparing the filters by taking these little pieces of, they're like this big and they're very expensive, and you put them in DI water and you get them soaking wet basically, and you get them all in a little beaker and you put them in an oven at 500 degrees Celsius to get them ready. And then you take an initial weight. And then once those are prepared, you put them in between these little, this is like my filtration system, you put them in between. And this is the water that we would gather on the sampling day. So I would put it in and then measure it, write it down, whatever it was. And then I would pour it into each one. So we would take three per tank. And they would come out looking like this. As you can see, actually a fun little thing in this one is that there is a tiny little jellyfish in there that you can't see, but it's the brown spot. So that's why there's a picture of it. I thought it was cool. And then, so after that, I would have to put them into the oven and they would wait there for 24 hours or until I could get to them. And that was 60 degrees Celsius. And then I'd weigh them and then put them back in the oven, but into a muffle oven, which is the 500 degrees Celsius oven, weigh them again, and then they were done. So 3 weights. And then, so that's what they would look like at the very end. And then on storm days, so these were this is like very specific to, there would be only 2 storm days throughout the whole experiment. So we would do the daily measurements like normal, and then we would do, but the difference was that Sean and I did dissolved oxygen every 30 minutes, which is time intense because you need to make sure that you do it right on time, because it's with the increase of the shear stress as it went up and the stepwise erosion that we were talking about. And then we took a water sample at the end of the day. As you can see, they all have different levels of resuspension in them. And then as Sean talked about with exportation, we would export 50% of the 3 tanks to make sure, like, so those are like the different tanks to make sure that those ones had the Cest removed immediately to see the differences. And as you can see, there's Elka. She sat there all day long on both of the storm days, making sure that computer was perfect. I don't know how she did it, but so I'll start with some of my results. So SECI, like I said, is about the light penetrating to the bottom of the water and seeing, which is actually 100, which may be a little tricky, but 100 meant that it reached the bottom. And that's something, and as you can see, it was pretty steady beforehand, but the 1 and the 2 represent our storms. And I'm going to remind you again, a storm is our recreation of a natural occurrence in nature, which is when we basically, it's just increasing the shear stress and making sure that it's just as natural as we can make it by studying the sediment in the water. And that's like our focus. So there's 1, and then as you can see, it dropped exponentially, but then the days after, actually all the water, like, it was clear for days on end. You could see the bottom of each tank all the time. And so we're really excited to see how the nutrients were affected in the sediment actually. And then storm 2, it happened again, and it even had a couple days where it was up top, but it was a little different because of that time that Sean talked about, the 1 week definitely changed the Cest and everything in the water, the mud. So at the end, you can see the non-export and export tanks started to be a little different in their finds. But okay, so the dissolved oxygen over the experiment. As you can see, it basically, once again, it was like a little up and down, just like the SECI was. But at storm 1, it went down. At storm 2, it also went down. So it's important to note, like, the storm is clearly causing an upset to the oxygen levels in the tanks. It even reached the point of hypoxic, which is an important word to know because that means that the levels in the oxygen are not livable for organisms in the water. And if it was like that consistently, most of them, if any of them, died for sure. And so it was just like that for one day, but it was that if it reached that level, that's very important to know because in the bay that's happening, it's a very real occurrence that's happening. And as you can see, the export and the non-export, there is a difference at the end. The export, and we have a theory about this. The theory is that because of the huge amounts of Ceston and TSS that were exported, there is a difference, like the oxygen was being taken up between. Okay, so for Ceston, as you can see, it's going, sorry, as you can see, it was really high. The TSS, the total suspended solids, Ceston, it was all really high during the storms, but pretty consistent otherwise. Same thing with POM. So particulate organic matter is what biodeposits are made of. That is huge to understand. That means if these levels rose during the storms, that meant those biodeposits, we can say for certain, were rising up into the water column, which is very interesting to study because not everyone gets to see that happen, because normally you just think they settle to the bottom. So then I would like to thank, before I start these next two slides, our collaborators over at Horn Point for the data on respiration in the water. So they really helped us out there. So as you can see with the increasing shear stress, the respiration in the water was also increasing. So that means that the oxygen was being used up as the shear stress increased. And then once again, that's happening with the TSS, the solids that are rising. The respiration was also rising. So like, why, you ask me, why is this important? So overall, that means if there's elevated TSS in the water, that is showing us that there's elevated respiration. Respiration is the act of the decomposing particulates in the water column. That means something is using those organisms, those microorganisms are using up oxygen. And if there's elevated oxygen use, that means the water quality overall is much worse, actually. So this data is very interesting find, because finding out like that connection between the elevated TSS and the water quality to be worse, it has not been done in a large experiment like this. And then effect on nutrients. We're sending stuff for further analysis, but should definitely be interesting to see if there's any nitrification going on in the water from the sediment that is at the bottom, and any light that reached down there, if that caused any like higher levels of things. So any of the samples that we took in the water will be in that TSS, which is what we're getting evaluated. So in conclusion, like I said, the additional biodeposits did affect resuspension because there was more Ceston and more particulate organic matter. There was respiration that was elevated during the storms, and we're getting further, there's going to be further analysis throughout the experiment. There's going to be further analysis with nutrients. Chlorophyll A, which is when the light reaches the sediment, there's like lighter mud. That's like chlorophyll A usually in there. And so that's what we're getting over here. This is like one of our last days with the tanks, where it's just us getting up for the further analysis to come. And I would like to acknowledge Sea Grant for their funding, and my partner, Sean. I love this picture of him holding a frog. The frogs were everywhere up there. They love the tanks, and so did butterflies and stuff. Sean wasn't kidding. We were in those tanks. We were super, super muddy. I love this picture. I would like to thank Elka so much. I love this picture of her with the giant tank. It shows you really how big they were. That's Mike from Horn Point. John for everything. I mean, he helped us so much. I think at least once a week we were calling John. And I would like to thank everyone that helped us put in the mud. The Oysters. Gotta give them a special shout out. And just everyone here in general. So thank you. Does anyone have any questions for me?
We do have 1 minute for questions. We have 1 minute for questions. Anyone on Zoom can unmute themselves or type in the chat.
I think Tom has a question. Yeah. Hello.
Somebody else have a question?
No, I think so.
So, thank you and Sean for your great presentations and all the hard work that you put into this. That was pretty evident. Wondering if you had any time to think about the results and think about with climate change expectations, we do expect more storms, more severe storms. What would you hypothesize would be the effect or effects of those more frequent storms based on your experiments? Can you draw any hypotheses?
That's a great question, actually, and I have thought about that, but I haven't sat long enough to really have a whole hypothesis. But I would think that it would just cause more upset to the bottom of like to the water, like all the water, all the organisms at the bottom of the water. They would constantly be resuspended and not have enough time to settle. And like if that's affecting the oysters, then that's obviously no good for our water quality. Our oysters are so important. And so if all of everything's being suspended all the time, and that means the water quality is worse, I think it's just overall a pretty negative viewpoint, sadly, if there's more and more storms going on.